And audience, sit and relax. Enjoy the session.
Yes, thank you. First of all, namaskaram, good morning, and welcome everybody. And second of all, I know it's a bit, maybe it's heavy for you, but ask any question if you have any time when I might talk, never hesitate, okay? If you do not understand, stop me. And that's the key. Also, other thing is that I can speak in many languages. So don't worry about English, just say whatever comes in your mind. Number 3 is that my main aim is to inspire you. Inspire you for something not like me, but million times better than me. Okay? And I come from a village in Uttar Pradesh, and when I was a child, I had no electricity, nothing in the house, and hard work and little bit of luck make me whatever I am today. So you have better than what I used to have. So therefore, you should do better than what I have, maybe a million times better, because you all have a cell phone. How many people have a cell phone? So you have, everybody has a cell phone. See how rich you are. And during my time, I had nothing. Okay? So I will talk about 2 things. Something, after sending you the title, I try to change it a bit so to make you more interesting, more engineering for you, okay? But my engineering is not that good as yours. And so I will talk about microbiome and health food. How many people eat yogurt? Everybody, it's very good. So something like that. And a cardiac stent. How many people knows about heart attack? Almost everybody, right? So these 2 things I will tell you. So I want to tell these to you that I co-founded with my students, master students, these companies, and I'm still involved there. But companies have their own lab, their own people, they're working. I am in the university, and I take part of them like a mentor, but I am as a president and co-founder of these companies. So if you ever want to get a training in a university, come to me. If you ever want to get a training in the company, come to me too. So both type of things. And also, for me, showing to you for 2 reasons. One more reason. A student like you can discover something that might be very useful for human. Okay? So that's one other inspiring point I want to give. Also, I want to show that no matter how small you are, you can try. And when you try, you can change it. Okay? So this is my research interest. A lot of different things. Okay? Like artificial cells, microbiome. And this is the name of the institute. I am a director of this center. Medical devices, drug deliveries, biomaterials, and cell and tissue engineering. So all these things we do in this place. Okay? Little complicated, but all these things we do for 2 reasons. Number 1, to develop something where people can use. That's number 1. Number 2, to understand and take the advantage of all the disciplines in one place. So my group, I have graduated maybe more than 100 students, master PhDs and things like that. So people from chemical engineering, mechanical engineering, physiology, medicine, microbiology, biochemistry, computer sciences, all they have worked with me in this place. And we try to develop few things where we can use it. You understand? Okay, so everybody understand what I'm talking about? Good. Yep. Why everybody is quiet? No quiet. I will not let you sleep. So microbiome, what I will talk about is, I will tell you about microbiome, what is a microbiome is, and I will tell you about what you have in your gut, so it is gut microbiome, and I will talk about probiotic health and potentials and limitations and conclusion. Everybody understand? So I will introduce you something new maybe, or maybe you know, and then how we can use it by giving one example. And I'll put you in a place where you feel that, okay, this is a real something that you could do. So let's talk about microbiome. This is a microbiome. What we have here around us, bacteria here, air is here, something here. Like all these things is around us, okay? And we have learned through our development in history to live with all these people, which is bacteria, viruses. You know, bacteria, viruses, everybody knows? Something that you eat, something you sneeze, sometimes you get a fever, sometimes I get all kind of problems, right? So all these things we have learned to live with it. So the few years back, 1995, when I was doing PhD, first time I proposed that a bacteria can be used to treat diseases. At that time, it was a kidney disease. So you have a bacteria you can eat by mouth, and instead of dialysis, you could have your urea and ammonia down in the blood. So the work of a dialysis machine can be done by a bacteria. That was my PhD topic. It was published in Nature Medicine, and it was a global release, press release, and things like that. So people are using now. So this is the kind of things I'm talking about. So this is economist have published a paper that we have so many bacteria in our body. Actually, in India, we have we think that we are made up of panch. So here we have actual fact, scientifically proven. Well, Indian is also proven, but here what we have is millions, billions of the bacteria around us. We have 10 times more bacteria than human cell. So actually we are not made up of cells, okay? Technically and scientifically we are made up of bacteria. So we are not XYZ, we are really bacteria, okay? So this is the economist saying it. I give you some idea. In the intestine itself, you have so many bacteria that whatever we eat, essentially they participate in there and they help you. Okay? But when the bacteria are become bad, they give you a disease, particularly when you are not eating good or you are come into contact with a person who is sick and you don't have an immune system developed around that particular bacteria. So we call them bad bacteria, right? Because it gives you disease. There are some bacteria that we have with us, we call them good bacteria. So this good bacteria is known as probiotics. Have you heard the term name probiotics? How many people you? Okay, good. So there is something, a bacteria you have in a powder or in a capsule, somewhere you use it to get the good health benefit. Okay? So that's called good bacteria. So I'll talk about microbiome and heart health, that how a bacteria can help heart. Okay? So let's talk about a little bit heart. Around 23 million people die every year about the heart, just because of the heart diseases. And India a lot. You know why? Because we eat a lot of coconut oil. And our artery genetically is not done properly. We have not developed that way. So in India, a lot of people, they have a heart attack. This is the fact. And there is one major reason for the heart attack is you have a cholesterol. Cholesterol is a chemical, a compound that is in the blood. Okay? If you have too much, you have a heart attack. If you have just one fact, that higher the cholesterol, higher the chances of getting a disease. So in United States, 99 million people have it. A lot of people. And every year, people are more diagnosed with the heart diseases. In fact, you can imagine this is by 2016, in last year, it's a $26 billion just the prescription in the United States alone. Globally, it's a huge prescription. You know what a prescription means? When you go to doctor, he gives you a perchi. Right? He writes which drug you take. So just for the managing cholesterol or lowering the cholesterol, people used $26 billion last year in the United States. So this is a good problem for everywhere. It's a huge problem. And it's a big problem. And in the US, because we eat a lot of fat, a lot of meat, and a lot of cheese, and a lot of bad diet in America and Canada. Here you eat very good diet. You don't eat that much. First of all, you eat this much, we eat this much. Okay? It's like a McDonald's and all kind of food that we have. There is that problem. And here we have, I think, is more eating ghee and coconut oils and some other lifestyle, because it's here people don't do exercise. Okay, things like that. So you have that. So everybody is with me? So far you understand what I'm trying to say? Good. If you're not, so how this cholesterol is come to in our body that creates so much a big problem? 85% of the cholesterol, our body make it. Nothing to do with the food. 15% comes from the food. Why our body make it? Because our body need it. For our brain we need it. For our every single thing in the body we need cholesterol, which is a good thing. So cholesterol is not a bad material, or it's not bad molecule, or it's not a bad something. We need it. Without this we cannot have a lot of things in our life. But once we have a higher cholesterol, then we have a problem. How problems happen? You are the engineer, right? So you have a pipe. In the pipe something is clogged. If something is clogged, you want to push the water, pipe bursts. Right? Same thing happen. You have arteries. In the arteries, once you have a high cholesterol, you have a plaque. So it gets deposited here. As a result, the blood going through here is not able to pass in a proper way. A lot of pressure, and then you have a problem. And the problem is like having a heart attack. So, since it's a big problem, there's a lot of companies are there to make money and to develop something new. Like a lot of things has been used. First thing is people try to do exercise and physiology, something like that. But you have a lot of drugs, many drugs. And these drugs, where they work? On two places. Number one, either they try to stop the biosynthesis of cholesterol, make our body full. So 85% that was coming from our genetics become less. And many of them, they work to stop that 15%. So we try to use the pathways in the human body that is involved either biosynthesis of cholesterol or making of cholesterol or degrading the cholesterol. So drug you develop to stop that. Everybody is following me? Yeah? No? Either you kill the problem by not having the problem, or you kill the problem by throwing the problem out. So in this case, either you want to stop the synthesis or you increase the use. Okay. So this is by 2030, it's $120 billion prescriptions in the US alone. So it's a real problem. And the drugs are very expensive, around $14,000 per year. And as a result, if I look at it, only 50% of the people in the United States are able to get the drug that they need because it's very expensive. In India, a lot of people even don't know they have a cholesterol because there is no proper every 6 months screening of the human body or if there's a problem, no screening. And the awareness is not there. People don't know. So, you understand? Yes or no? Okay, good. So it's a big problem. So there is something called bile salt. Okay? Bile salt is a chemical that our body synthesizes, and this is what we try to change so that we can develop something for cholesterol. So the bile salt is one of the steps responsible for biosynthesis of cholesterol. We're making of cholesterol. Okay? So we try to develop a bacteria that can eat this bile salt and this big molecule break into two small molecules. As a result, biosynthesis will stop. This one, we conceive with this idea that if this can happen, we will have not enough cholesterol in the body. As a result, people can use it as a drug. Okay? So how we did it? So we try to find out which bacteria has an enzyme that is good to break this bile salt. Enzyme is, imagine you have a hammer. How you want to have a hammer? Which one has a hammer powerful enough to break this? You understand? So we try to figure it out which bacteria can use. So we screen a lot of bacteria. In here is 350 strains, and come up with something that is safe for human use, because bacteria are bad, okay, and have a sufficient capacity to break that bile salt. You're there? You understand what I'm trying to say? So we develop that, and then we test it with a range of tests to see that they are safe, their genetics is good, they are able to survive when we give to the people in the gut, our intestine, and things like that. So all this, this is we come up with, and then we look at the genome analysis, that to see if there is any gene is there that can be harmful for human when we give them. Do you understand? So you want to develop something safe. So do that. You have to check all the way. So this is gene checking, how to and how it can be checked. And whatever I'm talking, try to write this one here if you want. That we have published everywhere, so we can see that. If you want to something more to study, okay? This is something interesting. Probably you will be. Something like this exists in the world only 3 places, okay, in the whole world. One is in my place. That's in my lab that I developed long time ago. So what is this? Is that this is a system through computer control program. This one is mimic how your gut is when you eat the food, where it goes and how it goes, how long it takes. So what is in your gut that is being affected? Imagine you drink a tea in the morning, right? You want to know what happened to that tea. So you cannot look at it if you have an animal. Or human, you cannot X-ray it. And if you want to look at it, that tea, how is it affecting your bacteria inside your gut, which is actually the making of your human, you need to have something, a device. So this one I spent a lot of money here to see that any bacteria that it changes, and not only that changes bacteria, we can look at their genetics level and enzyme levels. That how this bacteria is affected because of an antibiotics, because of a drug that you're eating, or because of food that you take, or even because the way you think. It's a total different science, okay? Very exciting. Even thinking can change your gut bacteria. Like yoga, people do it here and we think, we go quiet. Their bacterial cells, they behave differently because we have a different enzyme in our body. When you're quiet, we are angry, they are different. I'll show you some result. So we test there. Once we test there, then we test in the animals, whether it's a good bacteria or not. So how we do with the animals? Animals, there are a lot of animal available. So this is a picture showing of a hamster. Have you seen hamster in your house? No, eh? Okay. So what we have is a hamster, is a normal hamster. We buy them and we feed them, guess what? This is a coconut oil. Okay, anyway, so we feed them coconut oil, coconut palm tree oil, and as a result they develop high cholesterol. And we test them, we feed our bacteria and see whether they are able to lower it down or not. Okay, so this is a hamsters, 45 weeks, and we feed them bacteria and we measure them. This is the result showing up that hamsters that within 6, 8, and 10 weeks you are able to lower lot of cholesterol from origin by giving a single dose of bacteria per day. So you get excited. Oh, we discovered something. This is called discovery. Now you think about it, and then you saw something in animal is working. You're there? Everybody understand this? So you try to discover something, you go into the animal, try into the model like a GI model that I showed you, or in your lab on the cell lines or something, and then you try in the animals. So these are the things that you look at it when you look at the heart diseases, okay? So we look at all of them, and everything is going down because of that. Then we use a Richardson Center in Canada to do the human testing, see if it's good for human or not. So guess how I do it? So this place, we rented a hotel in the place. 150 people come. They live there for 115 people, and they came 6 months. Okay, so we give them food, breakfast, lunch, dinner, their sleep control, everything like something like a camp basically. And these people had a good high cholesterol. So we screen them, and then we do after something called randomize. You know randomize something? Okay. You're developing something, so you give to the people, you're eating it. You don't know what you're eating it. You're eating a water or you're eating a bacteria. Doctor who is giving, he don't know. And the people who are running the trial, we don't know. So nobody knows. So this is called randomized, double blinded. Blinded means everybody is closed. So nobody knows what you're eating. Just to see if the results are good or because of the thinking or because of something else. So there is no question asked that, okay, so that this is a not, yeah? Yes. Yes. So all these things, you have people, a lot of people comes, you screen them, you put them in the place that you want to. Some people eat placebo, means like a water with nothing inside. Okay? And some people eat the test drug, like a probiotics. But in the labeling, we label in a way that nobody knows exactly who is eating what. Neither you know, and I know. Nobody knows. Okay? So for these people, we start feeding them. They come in this, and we test all kind of things. So what we got is this. Their cholesterol went down the similar way that we saw in the animal. You understand? So we published that one too. So this is a result showing a different percentage of bad molecules that is happening in a human because of either genetically synthesis of cholesterol more or eating bad food, and that can be lowered down by a bacteria. Okay? Everybody understand? Yes? Good. Stop me if you cannot. This is what we other found, something called God discovery, give you something else. You're looking for cholesterol. We found something else. We found something that triglyceride for obesity. If the people have obese, like like me, you got obesity, we can lower that triglyceride in the blood. So this is the outcome of that. So then what we did is, okay, we can feed the people 2 weeks, 3 weeks, 6 weeks. We said, okay, 9 weeks, let's see what is going on. So this time, we hired again the similar people, and again it is a double blinded, so nobody knows. But this time in Germany, France, and Czechoslovakia. Okay, different centers. So we ship the material from Canada, and different centers they feed the people. And again they have their people. They come in the hotel. Their diet is restricted, everything, and try to figure it out exactly what we get. So this one was a very nice paper in Nature. We published it in clinical endocrinology. So this is a slide that I wanted to show you, very interesting. Every person who ate good drug, good probiotics, and every person who did not eat, this is the result of that. So every person during the feeding, whenever they try eating it, they take the drug, their cholesterol level was down. When we stopped them, their cholesterol level went back. You understand? So each person. So we can monitor it, each person. The people who did not take it, their cholesterol level was up all the time and no effect. So each individual. And then what we did is to look at it, that what is the range, because ultimately this is going to for each population where it is very mixed. Okay? So what we are saying here, this slide, this shadow is saying that yes, on the masses, if you give to whole India, for example, we will see the people will see whoever has cholesterol will go down. So this is the interpretation of results. It's not a direct result, but going to from 150 people to the whole 1.5 billion people. Okay? We saw something else. This is summary of the same result. First of all, we branded it. We call them Cardioviva, the name. We give a name that this is now a drug. Okay? This is a name. And what we found something else, that there are some other good molecule like ApoB is going down. So in this group, because we had a primary endpoint and secondary endpoints. Primary endpoints is something what you're looking at. And secondary endpoint, something you found it while looking at. You understand? When you do the experiment, you have A plus B, you want to say C. That's your idea. But sometimes you get A plus B, in between you get something else. So again, this is we got ApoB here.